Hi everyone. My name is Ivan Raikov. I'm actually a postdoc, not a professor yet at the Show Tesla, but thank you for the promotion. And thanks to the NCSA and the NSF for providing the amazing system that is Blue Waters that makes our research possible that wouldn't be possible otherwise. And today I will talk about our work on modeling the brain, well, a small part of the brain that we have been doing with the aid of Blue Waters. So first, the obligatory, I use Blue Waters to construct, simulate, analyze full-scale biophysical computational models of the rodent hippocampus and try to understand the role of the neural circuitry in processing spatial information. And so what do these words actually mean? Well, full-scale, we like to say that we do full-scale simulation because our models have a one-to-one correspondence of biological neurons to model neurons. And I'll mention why this is important for neuroscientists. And so our eventual completed model will have on the order of 2 million neurons and about 40 billion connections. So the work we've completed so far includes about half of these sizes. We called our model biophysical because we include detailed neuronal morphology, the dynamics of ion channels, synaptic connections, synaptic currents. And so every model neuron has on the orders of thousands of state variables. And finally, the hippocampus is a part of the brain that is thought to be responsible for learning, episodic memory, and spatial navigation. So a brief introduction about the methods that we use. So neurons are electrically excitable cells that have evolved for communication with other cells. They are characterized by these long membrane projections called neurites that are further distinguished into dendrites and axons. And their electrical activity is driven by a number of proteins on the membrane surface called ion channels, which act as transporters for the ions. They are usually, the membrane structures are reduced to these kind of segmented representations called compartments. And then the equations for each set of ion channels are solved via a partial differential equation solver. And the equations, they essentially reduce the system to these equivalent electrical circuits. So we use a solver called Neuron, which uses MPI and standard solver packages, and it's been developed over the course of many years. And then what has been understood about the brain navigational system is there's no complete operational description of how it actually works, but there's been a number of observations that we're trying to piece together in our models. So it has been observed that as an animal is running around in an environment, there's a number of neurons that seem to be active only at certain locations. These were called place cells. And they are essentially statistical observations over many trials. And furthermore, so these were first observed in the hippocampus, and then they were also found in other brain regions. And then a further discovery were the so-called grid cells, which were neurons that are active on a equally regularly spaced grid. They were found in part of the cortex. These discoveries led to the Nobel Prize in biology in 2014. But what's also interesting is that when people started measuring and looking at the activity of neurons associated with particular paths, is that they found all these place cells that are active in a particular sequence. These sequences were preserved over trials, and it was found that during the process of memory consolidation, when short-term memories are transferred from the hippocampus to the cortex, these sequences that were active during running are replayed very quickly. And so it is thought that this kind of process of repeating the sequences leads to consolidation. And from the point of view of individual neurons, usually these dots represent the times when the neurons emitted electrical pulses. So they are frequently model this Poisson processes. Then the intensity of the Poisson process is represented with these curves, and then the peak intensity is where the place field is considered to be. So that's the primary means of characterizing the system. However, most experimental observations can at best, so the very best experimental technology allows the observation of at most a few thousand neurons. And in the hippocampus of the rat, they are thought to be on the order of 2 million neurons. So clearly there's a very large discrepancy in scale. And currently we think that computational modeling is the only means to bridge that gap in experimental science and the actual living system. So the first project that we completed with the aid of Blue Waters was a model of the hippocampal CA1 region. So there's three major subfields of the hippocampus. That model included on the order of 300,000 cells, 300,000 neurons. There were a number of different cell species inside, so on the order of 10 species with their own biophysics. And they also followed realistic distance-dependent connectivity patterns. And in that model, we sought to resolve whether the hippocampus is intrinsically capable of the oscillations in the electrical field that have been observed in vivo. So there are particular rhythms called theta and gamma, which occur during running and during other activities. And a long-standing question has been whether these rhythms are imposed by other parts of the brain or whether the hippocampus is intrinsically capable of generating them. And they are important because they're closely connected with the sequence generation process. And so we constructed this model and then we subjected it to stochastic input with constant intensity. And then we discovered that the network is first of all intrinsically capable of generating these rhythms, but also it needed essentially all of these cell types. It needed a diverse circuitry because every neuron has its own time constant and phase preference. And so the combination of all these properties allowed the entire network to oscillate at this preferred frequency. And likewise, these are the high frequency oscillations that are thought to be associated with memory consolidation. However, the input provided to this network didn't carry any information content. We were primarily looking at the oscillatory properties. And in reality, there's a diverse set of information represented. As I said, there are place cells which are active at different locations. More recently, there was the discovery of cells that have been called episode cells or time cells, which are active at the beginning or the end of certain episodes of activity. Another discovery was the so-called predicting neurons that are active shortly before the animal is about to make a decision, such as turning left or turning right. So essentially the aim of our project, so all of these discoveries basically confirm that the hippocampus is intrinsically capable of generating sequences, and these sequences are capable of encoding both space and time. And so the goal of our projects is really to understand what are the essential properties of the hippocampal circuitry that allows these sequences to be generated and to encode spatio-temporal information. Yes. Okay. And so the reason why we actually want to use Blue Waters and model at full scale has to do with a lot with hippocampal anatomy. So this is the location of the hippocampus in the rat brain. And these are the names of the anatomical axes, so you can think of it as a long axis and a transverse axis. And this is the schematic of actually how the hippocampus is interconnected with the cortex. These are the three regions here. But what's interesting is that so the hippocampus has this approximate banana shape, and so we call this is the long axis and this is the short axis. But what's interesting is that as we move the long axis, there are projections coming from all these cortical and subcortical regions, as well as different regions can also output to a diverse set of output target regions. And because of the diverse kinds of information coming in from these regions, this actually led to also functional specialization. So even though the neurons contained here have approximately the same electrophysiological properties, actually they do have some functional specialization depending on the types of input. So traditionally, computational neuroscience had concern with just a small set of neurons, and the theory has been that the microcircuits are so stereotyped that you just need to understand the workings of this small set of neurons in order to understand the entire brain. But recent evidence really shows that because of these gradients of connectivity and functional properties, the full system is a very, very different beast than the microcircuit. So our focus for the past 2 years has been on the largest region of the hippocampus, called the dentate gyrus. This is again the microcircuit structure. The granule cells are the most numerous. They are thought to carry the information content. These other interneurons, they are thought to perform gain control, so they regulate activity as it changes with new sensory stimuli. And most importantly here, we aim to really create a realistic representation of the spatial information that comes into the hippocampus from the cortex. An important aspect of our work has been to create a realistic anatomical representation of the entire hippocampal volume. This is a parametric surface generated from anatomical reconstructions and cell bodies distributed inside the volume. We also basically have generated neuronal dendrites inside that volume following anatomical measurements. So this is a transverse slice that shows how really densely packed the neurons are. And what's fascinating is that these dendrites, they all receive highly overlapping inputs, yet the cells actually are able to distinguish between very small differences of inputs and code the information very sparsely. And finally, in order to generate our connectivity, we actually need to follow the anatomical gradients rather than just 3D Euclidean space. So we created sort of a projection of the 3D locations onto a 2D sheet according to the relative distances, the relative sizes of the layers in each Z position. And this resulted in this kind of skewed connectivity, which actually turns out to be equivalent to an attractor network. So there's very clear evidence that the anatomy actually has very particular functional consequences for these models. And then finally, this kind of anatomical structure allows us to define input structure to the hippocampus that follows the experimentally observed gradients. This is one of the papers that led to the 2014 Nobel Prize, observed that at different locations in the hippocampus, the types of cells that project to it have different spacings and different sizes of their place fields. So this is our rendition of these gradients. Essentially, blue is smaller spaced spatial inputs, red is larger spaced spatial inputs. There's a number of cell type specific projections within the hippocampal network. Here is, for example, a cell that tends to project over very long distances, over 5 millimeters. So we couldn't have really expressed these relationships in a reduced scale network. This is the activity of the network, where every dot is a neuronal spike. And actually, to make sense out of this, we need to convert these to firing rates, which correspond to the Poisson process intensity. And so on the left is the spatial input, which is highly overlapping, so many cells carry the same information. On the right is very faint, unfortunately, but it's a very sparse but accurate encoding of this same spatial information. So this shows that our model is capable of the coding information in a very highly sparse manner, which is thought to be a critical property of the hippocampus. And this is the process of pattern separation. So for highly overlapping inputs, we want to essentially have a non-overlapping spatial representation. This is a quantification of this pattern separation. So this is the overlap of inputs, or the correlation of inputs coming into the dentate, into our model. And these are the principal output cells, which show very low correlation. And this is another representation of the same thing, just indicating this is really for the first time that we're able to show this in a full-scale biophysical model. And finally, the next step for us is then to build, so this is our existing dentate gyrus model. The next region of the hippocampus we're aiming to model is CA3. There is a fairly complicated interplay between those two regions, and there's a prominent theory that essentially states that the CA3 modulates the generation of sequences. So we are now working towards modeling this. In conclusions, we have models comprised of diverse cell type-specific connectivity, anatomical distributions of cells. We have to do this in order to accurately represent spatial information processing in the hippocampus. And so far, only Blue Waters has had the capacity to do this. These are some of the runtimes of our model. So just 10 seconds of activity still takes a few hours on 2048 or 4096 nodes. Beyond that, our scaling is no longer linear, so we haven't tried larger sizes. And finally, this is our lab at Stanford, external collaborators, and I wish again to thank Blue Waters and NSF, and NIH provides most of our research funding. So thank you all.